Today I'm going to share some information about single-cell multiomic technology that reviews extensive influence of genome structure on gene expression. My name is Sheng Zhong. I'm at the University of California, San Diego. So the key question that many folks would like to address is whether chromatin architecture exerts a widespread impact on the regulation of numerous genes. So this remains an open question, perhaps even a debatable question at this time. So one of the ideas to attack this question is proposed several years ago to leverage single-cell multimodal omics technology to assay simultaneously the genome architecture and perhaps gene expression to obtain new information that perhaps relevant to this key question. So some of the recent developments have been able to simultaneously obtain the information of genome organization. Here I'm showing a partial list of the recent technologies, especially on the imaging front. This is a partial list of several technologies, including DNA-seq FISH plus ORCA MINA that can obtain chromatin traces together with gene expression. On the omics front, so there are gauge-seq, HiRes, and MuSeq that could simultaneously interrogate co-complexed DNA fragments and gene expression. So I would like to bring your attention to MuSeq technique that I will elaborate. So this technology would not only obtain pairwise genomic interactions, but also multi-way genomic interactions. It will interrogate from the same cell gene expression as well as RNA chromatin association information. So MuSeq stands for multi-nucleic acid interaction mapping in single cell. So the rationale for us to develop such a technique is that a single chromatin complex can be dynamically organized to harbor multiplex chromatin interactions that could include chromatin-associated RNA. So our design goal of this technology is to enable simultaneous profiling of gene expression, co-complexed DNA sequences, and RNA chromatin associations in the same nucleus. Specifically, we would like to construct a single DNA sequencing library from which we wish to sequence RNA and the fragmented DNA sequences together, tell which RNA sequences and DNA sequences came from the same nucleus, tell from the sequencing data whether the source molecule was RNA or DNA, identify any DNA-DNA and or RNA-DNA associations, including multi-way contacts. So the way that our lab would like to approach this was originally proposed by Tri Nguyen, now a president fellow at Stanford University, and later completed by Dr. Zhifei Luo. So this MuSeq workflow starts with adding a DNA linker or an RNA linker specifically to the DNA or RNA inside the nucleus, and then use a split pool strategy to add nucleus-specific barcodes to every fragmented DNA sequence or RNA sequence, and then release the nuclear complexes from the nucleus and use this microfluid system to add complex specific barcodes, and then use another round of split pool strategy to add additional complex specific barcodes. So in the end, we would like to construct a library in the following form. This DNA sequencing library could have contained either an RNA insert or a DNA insert The way that we can differentiate the RNA insert and the DNA insert is by its neighboring linker sequence that was pre-designed. So if we read an RNA linker sequence, we would tell it's RNA insert, and otherwise it's a DNA insert. And then using the combination of cell barcodes added by split pool and complex barcodes added by the 10x microfluidic device assisted barcode addition, together we could differentiate, identify single molecular complexes from within single cells. So to test if music would generate data that would comparable to many other data types, we assembled single cell music data and compared them to micro-C derived chromatin contact information here on the right. Sheng Zhong led all the data analysis. So here we see that the bulk level chromatin interaction information is to a large extent comparable to the ensemble of single cell music derived chromatin interaction information. So if we go to single cell gene expression at the bulk level on the y-axis, we see that the bulk nuclear RNA sequencing derived gene expression levels is comparable to the ensemble music generated single cell generated RNA expression level on the x-axis. If we go to a specific locus on the right, we see that from the first track, there's bulk RNA sequencing data. On the second track, this is bulk nuclear RNA sequencing data. And from the third track, this is a ensemble of single cell music data. And the next three tracks are the single cell RNA transcripts. And from the minor strand, the blue tracks, we see again the bulk from RNA-seq, the bulk from the nucleus, and music ensemble, as well as each single cell transcripts. So from here, we can see that at the transcript level, music can generate fairly reliably transcript measurements from single cells and differentiate the original strand from which the RNA transcript come from. So if we use an imprinting gene locus here on the right, we see that from the paternal and maternal alleles, in this particular case, the DNA-DNA contacts are comparable, whereas the RNA-DNA contacts was specific coming from this imprinting transcript, imprinting gene, KCNQ1OT1, that was only detectable from the paternal strand, and the RNA chromatin association was only detectable from the paternal strand as well. And so to quickly summarize, here we see that music concurrently profiles DNA-DNA, RNA-DNA interactions, and transcriptome in single cells. The ensemble of music single cell data recapitulate the expected patterns of the 3D genome structure, gene expression, and RNA-DNA association. So leveraging this technique, we profiled 14 human prefrontal cortex samples, including 7 male and 7 female samples, generating a single cell map of gene expression and RNA-DNA complexes in human brain. So these together resolved more than 9,000 single cells. And if we plot these single cells by their transcript abundance level, we see that all the major cell types from human cortex can be resolved. And more than that, we can dissect some detailed cell type information within these major cell groups, including some of the cellular states within microglial cells, for example, and some of the detailed cell type information from excitatory inhibitory neurons, for another example. So one of the recent open question is from the observation that the cis expression quantitative trait loci, cis-QTL, often but cell type specificity. So these cell cis expression quantitative trait loci, cis-EQTL, are genomic sites where personal genomic sequence variation explain the expression variability of a target gene. From a recent Nature Neuroscience study, they identified approximately 500,000 cis-EQTLs in the human brain. So on the right, we use this offset plot to summarize this previously identified cis-EQTL and target gene pairing information. We see that most of the cis-EQTL and target gene pairs are specific to a specific cell type. For example, excitatory neuron, inhibitory neuron, astrocytes, oligodendrocytes, or microglial cells. There's, relatively speaking, very few of these EQTL target gene pairs are shared across multiple cell types. So this suggests that most cis-EQTLs influence the expression of their target genes within particular cell types. So this observation also provoke a question. What molecular mechanism makes EQTL affect target gene expression in a cell type specific manner? Think about it. Across a person, the multiple cells in general share the same genome sequence. Nevertheless, a specific EQTL locus would only manifest a personal gene expression variation of this individual to other individuals in specific cell type, but not in other cell types. So what might be a mechanism, and whether there is a general mechanism behind this specificity? So to investigate this question, we started with two specific cases. Here we investigated a case of two cis-EQTL in excitatory neuron that affect the expression, the personal expression variation of the NEL1 gene in excitatory neuron. And also, we investigated another case, which is a PREX2 gene. There are 6 cis-EQTLs in oligodendrocytes that would only affect the personal variation of the PREX2 gene in oligodendrocytes, summarized on the plot on top right panel. So now with the music data, we can plot in every single cell and summarize by cell type the chromatin contacts between the sequences covering the promoter sequence of the NEL1 gene and the sequences that harbor the two cis-EQTL, showing in blue arrows on the right. We see that all the measurable chromatin interactions between the promoter sequence of NEL1 and the cis-EQTL of NEL1 were from the excitatory neurons, not from the other cell types. So these cell type specific chromatin contact coincide with the cell type specificity of cis-EQTL and target gene pairing. In the other case, the PREX2 case, we see that all the measured chromatin contacts between cis-EQTLs of the PREX2 gene and the promoter of the PREX2 gene are within oligodendrocytes. There was no measured contacts in the other cell types from this human cortex, from all these human cortex samples. Again, in this time, the chromatin contacts linking the cis-EQTL and the promoter sequence are from the same cell type where the cis-EQTL and target gene pairing happened. So in these two cases, we see this coincidence, and we wanted to ask whether these observations can be generalized to most EQTL and target gene pairs. To do this, we categorize all the cis-EQTL and target gene pairs by their cell types in the columns in this figure. And we categorize all the chromatin interactions by the cell type that they are measured as a rows in this confusion table. From this large confusion table, we can do association test between the eQTL target gene pair and chromatin interactions within each cell type and outside each cell type. So these chromatin association tests yielded association metrics on the right. These are odds ratio and also confidence interval and significance level of odds ratios measuring the association. And all these odds ratios are greater than 1 and significant, suggesting that in all these cell types, there is an enrichment of the cell type specific cis-eQTL target gene pairing and cell type specific chromatin interactions. So this suggests that the observed eQTL target gene pair in a cell type is not independent of their chromatin interaction. And this is a general phenomenon across all the cell types, across all the obtained eQTL target pairs. To quickly summarize, here we see that cis-eQTL often affect gene expression in specific cell types, and eQTL target gene pairing correlates with DNA-DNA contacts in the corresponding cell type. This suggests a widespread impact of 3D genome structure on the expression of numerous genes. So this single-cell music data also allow us to interrogate the chromatin organization in every single cell, plotted as columns in the heat map on the right. On the right, every column represents a single cell. Because we have measured the transcriptome of each single cell, we can easily categorize the single cells by their cell types. And the color intensity on the y-axis indicates the chromatin conformation contact frequency. Taking the first few columns, for example, in the excitatory neuron, we see that for most single cells, most columns, the most frequent contact frequencies are from the DNA sequences separated by relatively short distances at the bottom. So for example, between 10 kb and 100 kb regions, most single cell exhibit the most frequent contact frequencies. And the longer the genomic distances, the fewer chromatin contacts there are. While a large fraction of the single cells conform to that expected pattern, again, that expected pattern is the closer the genomic distance, the more chromatin contacts. We observe, surprisingly, that there is another subset of single cells which lost the compact local chromatin structure. We see that in the purple group, this group of single cells no longer have their most frequent contacts necessarily concentrated in the genomic sequences that are nearby. So we call these two subsets of single cells as local chromatin structure preserved single cells, LCS preserved in short, on the right in pink. The other subset as local chromatin structure eroded, LCS eroded, on the left. So what are the two subsets of single cells mean physiologically? Leveraging their single cell transcriptome, we could tell that the group on the right in pink tend to have younger transcriptomic age. And the group of single cells on the right tend to have older transcriptomic age. Furthermore, if we map the AD Alzheimer's disease pathology, we see that the group of single cells on the right that exhibit the preserved local chromatin pattern tend to enrich with normal pathology, whereas the subset of single cells on the left tend to enrich with Alzheimer's pathology. If we analyze the two sexes, we see that in this particular case, the males are enriched with the preserved local chromatin signature, whereas the eroded chromatin signature tend to be enriched in the female. So to summarize, we see chromatin structure reflects single cell age and disease status. Brain cells exhibit variable degree of loss of a compact local chromatin structure. These LCS eroded cells exhibit an older transcriptomic signature. Alzheimer's brain has more LCS eroded cells, namely older cells, than control brain. In oligodendrocytes, female has more LCS eroded older cells than the male. So I've mentioned the leaders of this project along the way. In addition, I wish to also mention Wenxin Zhao, Sherry Wan, John Richard, who also contributed significantly to this project. And we have a number of important collaborators and colleagues at UCSD and other universities, many of whom are key researchers in the 40 Nucleome Consortium. I also wish to acknowledge the grants that are supporting this work. With that, I would like to open for any questions you may have.